Hello, everyone. Welcome to the Water Research Foundation webcast on Project 5154, Autonomous In Situ Monitoring of Harmful Algal Blooms. We're so excited that you're able to join us today. My name is Sydney Samples. I'm a research manager at the Water Research Foundation, and I will be moderating today's webcast. We have a fantastic speaker lined up today. You'll be hearing from Dr. Amit Pinto. Dr. Pinto is an environmental engineer and Carlton S. Wilder Associate Professor in Civil and Environmental Engineering at Georgia Institute of Technology, or Georgia Tech. Amit is a chemical engineer from the Institute of Chemical Technology at the University of Mumbai, with postgraduate degrees in environmental engineering from the University of Alaska and Virginia Tech. Their research has received support and recognition through prestigious grants and awards like EPSRC's Bright Ideas Award in 2015 in the UK, the NSF Career and ISME IWA Rising Star Award in 2018, the Paul L. Bush Award for Innovation in Applied Water Quality Research in 2019 from the Water Research Foundation, and the IWA MEWE Mid-Career Award in 2023. They were inducted as a Fellow of the International Water Association in 2024. Dr. Pinto's research focuses on the development and application of the state-of-the-art molecular and modeling tools to monitor and manage the microbiology of natural and engineered water systems. So we're going to have a really great presentation for you. But before we get into that, a little bit of housekeeping. So throughout today's presentation, you can type your questions in the box below the presentation on your screen. We'll answer as many questions as time permits at the very end. So please submit questions for us. We want to hear from you. Slides and a recording of today's webcast will be available on our main website. You can download the slides now, as well as access a link to the project's page with the final report for this project. So please check that out after today's webcast. A certificate of completion will be automatically generated after the webcast. If you have any questions, please contact our webcast coordinator, Michelle Suazo, at the email provided here. And then lastly, please stay tuned at the end to fill out a quick survey so that we can make sure our webcasts are best tailored to your organization. So with that, I would like to pass the floor to Dr. Pinto to present on 5154. Dr. Pinto, the floor is yours.
Thank you so much, Sydney. Thank you for the introduction, and it's a real honor and pleasure to be here presenting some of the work that we did as part of a tailored collaboration project 5154. And the project is titled Autonomous Monitoring of Harmful Algal Blooms. While the title slide just includes my name, this project has a fantastic team that spans academia, industry, and what I want to do is first thank the entire team of this project. At Georgia Tech, I was the PI, and this project was really led by two graduate students in my research group, Mr. Farhan Khan and Benjamin Ginsley. We were very fortunate that we were able to collaborate, and we continue collaborating with a fantastic research team at the Great Lakes Water Authority led by Dr. John Norton. This was a tailored collaboration project, so GLWA contributed to the cost of this project. This project involved collaboration with an expert in deep learning and artificial intelligence, Dr. Miguel Fuentes Cabrera. While he was at the Oak Ridge National Laboratory, he has since moved to Northeastern University. And then I would also especially like to acknowledge Dr. R. Michael McKay and Emily Varga at the University of Windsor. And Dr. McKay has decades of experience working in the cyanobacterial and HAB and freshwater monitoring landscape. And without his team's expertise and without his team's contribution in terms of helping us make sense of what we were seeing, the outcomes of this project would not have been possible. We are also very fortunate to collaborate with Limnotech, with Ed Verhamy and Tad Slavicki, who are collaborators on this project. So that is a fantastic project team. It was a pleasure to work with everyone involved, and at the same time, I also want to acknowledge The project advisory team, those of us who have been working with the Water Research Foundation, collaborating with the Water Research Foundation, know how important project advisory committees are. Zaid Chaudhry, Lisa Ballantyne, Trista Brophy, and Atlasi Daneshwar. This is, we are very grateful for the time that they devoted and dedicated to this project, and providing extremely helpful feedback that helped us improve the outcomes of the overall project. Now, having said that, let me get into the technical content. What I want to do in the first few slides is map out, obviously this presentation is about monitoring of harmful algal blooms. And when I when I'm talking about harmful algal blooms, what we're also seeing, what we're interested in is monitoring the microbiology of fresh waters, of source waters that are raw water for our drinking water utilities. Since we're talking about autonomous in situ monitoring, it's good to get a sense of what is in the landscape in terms of HAB monitoring. So I'll orient you really quickly. What you're seeing over on the y-axis is the purchase cost of the infrastructure required to do monitoring. And on the x-axis is the technological readiness level. So higher the TRL, the more close it is to being applied in the field. And the color of each of these technologies that shows up over there goes from gray to red to yellow to blue, and that depends on how easy or difficult it is to use. So if it's something is non-technical usability, then it is high level. So people without very specific expertise can use that technology to perform that monitoring. And something that is on the gray side of things, you really need a lot of technology. And the symbols over here that you see on the right over here, the symbol is field, means where that technology is based. So here you have 4 examples of different approaches that can be used for harmful algal bloom monitoring, and these are all what can be applied in the field. So from multi-channel fluorometers to dipstick to HAB scope, and relatively high technology level. And dipsticks are pretty inexpensive. I am now showing you benchtop instrumentation that can be used for monitoring of harmful algal blooms. You have molecular methods such as isothermal amplification, you have ELISAs, and then you also have a fantastic instrumentation called FlowCam, which is a flow imaging device, and it is on the expensive side, but extremely high technology readiness level in terms of use. So these are benchtop instrumentation. Then you have moored instrumentation, something that can be deployed in the field, but requires some supporting infrastructure. The cost of these instrumentation tends to be relatively high. And finally, you can also use satellite data, whether that's hyperspectral remote sensing, whether that's multispectral remote sensing. You can use this data to get a sense of what's happening with the microbiology of these freshwater systems. So I'm showing you what's kind of on the market these days. I'm sure there is lots of other wonderful stuff out there as well. But when we were exploring this space, one of the things that we realized that we had been developing a technology now for several years that could actually be used and it could be useful to the field and to drinking water utilities, irrespective of size, or sort of irrespective of sort of the price point, to perform phytoplankton cyanobacterial monitoring of freshwater systems. So what is this technology that I'm talking about? So over many years, and one of the things that I should first credit over here is the Paul Bush Award. The Paul Bush Award really laid the seed funding that helped us get started to develop this technology. And since then, we've been fortunate enough to work with fantastic collaborators at University of Illinois and University of Buffalo, and be supported by the Bioenergy Technology Office of the Department of Energy to develop this instrumentation further. And the application, and so in that little box, you can see 2 little devices sitting over there. These are what we call the Autonomous Real-Time Microbial Scope, or the ARTiMiS. So I'll be referring to this instrumentation as the ARTiMiS moving forward. And the ARTiMiS is, as you can see, it's a very tiny device. And I'll give the size specifications later. And what we had done with this ARTiMiS over the last many years was really get it to the point where it could monitor. What you're seeing over here is photobioreactors that are used for nutrient removal and recovery at wastewater treatment plants, and at the same time producing biomass, sort of microalgal biomass that can be used for certain end products. And this is Ben over here in the field, and here you can see the photobioreactors and ARTiMiS in operation as well. So when we were exploring the space in terms of cyanobacterial HAB monitoring, we thought that maybe the ARTiMiS could contribute over here. So now, what is the ARTiMiS? So the ARTiMiS is an autonomous multimodal imaging platform that essentially leverages a suite of deep learning tools to identify, classify, and count zoo and phytoplankton species in near real time. So all you have to do is you can put a sample. Here you're seeing a little sample of from microalgal biomass from the photobioreactor system. This little pump loads it into the instrumentation. The instrumentation images the sample that is inside it in multiple different modes of microscopy. And what's nice about this instrumentation further is that it not only takes the images, but it's also processing these images on board. So this is what the ARTiMiS is. It's a sort of self-contained, entirely autonomous little device that can image cells and spit out data to tell you who is in the image that, or rather what is in the image that it captured. Now, a few more details about the ARTiMiS. So we build it in our lab for less than $1,000. The bill of materials is quite inexpensive. We fabricate it. It uses non-traditional optics. What's also quite nice about it is that it uses an embedded single board computer. That means you don't need any other hardware outside of it to kind of run and process your samples. It is a very compact design. It's only about 17 centimeters by 17 centimeters, and we are working to make it even more compact. What's also quite wonderful about this is you can actually run this off a battery. You don't need, it doesn't need to be plugged into a lab. So it also makes it quite portable. It doesn't need any consumables. Now that's a very powerful thing, because if it doesn't need any consumables such as dyes or stains, it's important because now you're not producing any chemical hazardous waste out in the field, right? So the sample that you pull into the ARTiMiS for image, you can discharge back into the environment because essentially you've not added anything to it except shine a light on it. This is an important point. What this also reduces is in-field maintenance and someone to go out into the field and fill those chemicals or consumables. You don't need that with the ARTiMiS. And the last aspect of it is that it is very, very easy to maintain. And I won't get into a lot of this. Is that if there is a lot of interest in this, I'll talk about it later. But it's very, so the take-home message is it's a low-cost, portable, self-contained device that does not need any consumables to do what it needs to do. No consumables, no extra hardware. And samples can be run directly on the ARTiMiS. So you don't need to do any processing for the samples at all. You can take an aquatic sample and just put it in the ARTiMiS and it'll run those samples for you. What the ARTiMiS does do is it generates high resolution images of microalgal species. So these were images that we captured from these photobioreactor systems. And you can see, and this is an important point that I want to bring up as I move forward. What you're seeing in these images in this photobioreactor system is relatively low diversity. Now, the reason this is relatively low diverse is because these are microalgal mixed cultures that are grown in reactors under very, very specific conditions. So what that means is that it is selecting for certain types of populations, just like in a typical wastewater treatment plant, where we select for certain types of populations for the functions that we want. This point will become important as we think about translating what we developed to monitor photobioreactor systems, and we translate that out into the field. And you can imagine how many things are now going to change as we take this instrument and move it out into the field. Built onto the ARTiMiS is also a classification approach, and the classification approach leverages a convolutional neural net and it achieves very high levels of accuracy. What you're seeing over here is what we call a confusion matrix. On the y-axis is ground truth label. That means this is a label assigned to a particular image by a human expert. And on the x-axis are predicted labels. What did the machine learning algorithm classify it as at? And as the colors in the diagonal become darker, the accuracy becomes higher. And here you can see on the top, the overall accuracy of the ARTiMIS is about 92% for these microalgal cultivation systems. So that's absolutely not as bad at all, especially given the particular price point. What's also fascinating about this in this particular image is that you're seeing the same species of microalgae in different morphological features. So you are seeing Senedesmus in clusters of 4, clusters of 2, or clusters of 8. The ARTiMIS can also distinguish within a particular taxa for different morphotypes. And this becomes quite a powerful feature, because now we are talking about being able to predict some aspect of the physiology of a microbial community or microbial community member in its environment. So this is about what we had done with the ARTiMIS leading up to this tailored collaboration project. And since then, it's improved quite a bit as well on the application space in terms of photobioreactor monitoring. So question for you, or question for us was, how could ARTiMIS fit in this HAB monitoring landscape? We knew that we had a technology over here that could really add value, give a new tool, a very useful tool for our water utilities or a host of other stakeholders to kind of apply for cyanobacterial monitoring. The first thing we knew is, relative to everything else on the market, ARTiMIS fell into this particular price point. It's not as cheap as dipsticks for sure, but it's also a lot cheaper than ELISAs or a multi-channel fluorometer or what you need for example, PCR amplification. So it's quite inexpensive. We knew that it was there. What we also knew is that while we had developed the ARTiMIS and it was working quite well for these photobioreactor systems, we knew that it was not quite yet ready to be applied in the field, because there were several challenges that we anticipated, and I'm going to talk through those. The bulk of the presentation is going to be about those challenges and why those challenges are important to solve for any instrumentation that wants to use an optical approach. So we knew the ARTiMIS wasn't ready. We couldn't just put it out in the field by a lake and monitor what's happening in the lake. So what the 5154 project, the aspirations of the 5154 project, was to take the ARTiMIS to where we anticipated in terms of technology readiness level of maybe 2 to 4, somewhere in that space, and take it to a technology readiness level of 7. So not yet quite field ready, but almost there. And I will report the progress that we made in the rest of the presentation, and we can assess what the road ahead looks like, what else kind of needs to be done. So the overall vision of the project was to take our device and get it to a point where it could do autonomous real-time monitoring of freshwater ecosystems for early detection of harmful algal blooms. So we knew that because of the price point where this device sits, if we are able to get it out into the field and make it work autonomously, you could have multiple of these devices across a watershed autonomously monitoring, and potentially at some point in time, if there is an issue, alerting the operators, alerting water utilities. So the goal of this project was to adapt the ARTiMIS device for real-time monitoring of HABs in the raw water intakes of Great Lakes Water Authority. And this is where we found a fantastic partner in Great Lakes Water Authority. And they were quite interested because some of their raw water intakes are right here in the Detroit River. And the Detroit River can be influenced by Lake St. Clair over here, from water flowing from Lake St. Clair into Lake Erie. And there have been instances of HABs in Lake St. Clair, and we obviously know about Lake Erie. So we collected samples from these 4 locations that you're seeing over here. And we also collected sample from Lake Huron, this intake for the Lake Huron water plant, as kind of a control, because that water is really, really quite pristine. And again, before I proceed, I'll just highlight over here, a lot of the data that we analyzed comes from these samples that were collected. So these are not some sort of lab constructed samples. These are field relevant samples. This is what the water looked like. This is what the fresh water looked like at these locations, at the raw water intake for drinking water treatment plants. And again, I have to come back and thank the project team, because we also cultured some specific cyanobacterial species of concern to the Great Lakes region. And we are very grateful to Dr. McKay for helping us acquire these cultures, and for his group's sort of know-how on how to grow specific type of cyanobacterial species. Now, I talked this far about how great this device does for microalgal monitoring and photobioreactor systems, but that going into this project, we knew that we would have to place this instrumentation at a relatively low technology readiness level because of the challenges that we were going to encounter. So what were the challenges that we did encounter? So these are 5 challenges that we had not yet seen in those microalgal cultivation systems. The first challenge is that, unlike in a photobioreactor, where there are high selection pressures, in an open freshwater environment, particle size varies significantly. So this is an important point, and I will show you why that matters so much. The second aspect, particle size varies quite a bit. The second aspect is because the concentration changes in freshwater systems depending on season. What's essentially happening is you have times of the year when you have low phytoplankton density. And when those concentration decreases, I'm going to call them stuck particles or background particles, but I'll get to what I mean by that. But when the biomass concentration decreases, you start running into some issues with debris or with how outliers really affecting your ability to get quantitative estimates. The third is, I'm using these deep learning models, but is my model really using the data that I want it to use? Again, we'll show you an example. Can the ARTiMiS process the data that we're gathering? Like I said, we want to get to a point where there is not another hardware that you need, and the ARTiMiS does everything. And finally, do we know everything? There's this, there are known knowns, there are known unknowns, there are unknown knowns, and there are unknown unknowns, right? There are all these 4 categories that we have to deal with. So how do we deal with that? So let's get to the first point, high variation in particle size. So the way the ARTiMiS works is it takes an image, and you're seeing an example image over here, and the image processing algorithm looks for edges of objects in that space, and then uses what we call as an object detection algorithm to extract that object from that image. What you're seeing over here is the ARTiMiS has identified all of these different objects, and it has put those yellow bounding boxes on it. And now it's going to extract those objects from those yellow bounding boxes, and it's going to analyze them. What's nice in this particular image is the variation in particle size. There is some variation, it's not too high. And at the same time, the biomass concentration is low enough where there is not a lot of overlap between different objects. Now, this is particularly not common when you're having a bloom. When you're having a bloom, what you're seeing is very high particle variation. So over here in this plot, what you're seeing is microcystis in samples that we collected. And on the y-axis, the size of the microcystis colony. You're seeing that the size of the microcystis colony can vary over 5 orders of magnitude. That's huge, right? And so if we take an algorithm that we had designed for those photobioreactor systems and we apply it to extract a microcystis colony, you can see those bounding boxes are actually splitting up objects, creating artificial objects. It's just not as clean anymore. That big microcystis blob is now being split into multiple different aspects. So what we had to do as part of 5154 is actually experiment quite extensively to identify what are the different types of models What are the different types of models that are going to help us overcome this problem of high variation in particle size and overcrowding in these? So what we have developed is an algorithm called a segmenter, and it doesn't work 100% well, but it works much better than what's out there right now, where now you can see that it is actually capturing the entire microcystis colony in a single object. Now, what that means is we can observe and analyze this entire microcystis colony as a whole, and not simply as different particles. So this is important, right? A human eye can do it, but doing this autonomously is not that straightforward. So this was one of really an important accomplishment of this particular project, is for this instrumentation to sort of on a relatively sort of low cost hardware and compute power to be able to extract objects and analyze this. So we developed an optimized object detector to detect and extract diverse objects. And what this allowed us to do is start collecting now good quality images. And these are some example images that we've collected from during the course of this particular project, from samples that we received from the Great Lakes region. And why is it so important to remove objects in individual patches? And the reason is, at least at the beginning, we need to be able to assign a taxonomic label to these individual objects, and then use those taxonomic labels to train a machine learning algorithm. So that's essentially what we did. We created, over here you can see 12 different classes. We created large annotated image libraries. This was quite painstaking work for my graduate students, and in this process we also trained a lot of graduate students and undergraduate students in taxonomy of some of the stuff that we were finding in the Great Lakes region. But creating these image libraries is absolutely very, very important, because honestly, these machine learning algorithms, these convolutional neural nets, for example, will only do a good job of predicting what they see or what they have seen as part of the training data set. And this is an important point, and I'll get back to it in a little bit. So problem solved, high variation in particle size. We've solved this problem with an object detection algorithm. We can create lots and lots of libraries. But what we ran into as part of this project was this tiny little issue. Oh, my video is not showing over here. Is there a way to fix this? I had an embedded video. If not, that's okay. I can.
The video probably won't work.
Video won't work. Okay, that's okay. But I have a screenshot of this as well in the next slide. No problem. So the challenge that happens is when you have a low biomass environment and you have a little chip that's collecting a sample, pushing a sample through it, taking an image, and then flushing that chip for the next sample to come in, you often get tiny particles from the previous sample getting stuck. So over here, if the video had played, you would have seen this. Over here, the yellow boxes without the red circles are fresh objects that the ARTiMIS has seen. And the red circles are objects that are stuck over from the previous sample, right? Because every sample only takes a minute or minute and a half to process. You can actually take like 15, 20 samples an hour. So you can have it just pretty much just running on its own. And so you can imagine the challenge that happens now, right? If objects are getting stuck, they are being double counted, and suddenly the concentration in this particular sample is 10%, 20% higher than it really is. And this is a problem that we kind of need to solve. We had not seen this in the high biomass environment of the photobioreactor system, but this is a real problem. And in the course of this, we realized this is not just a problem over here. This is a problem in many fields of microscopy where this happens. What's also quite interesting, now you might imagine, why not just identify those objects and toss them out? Maybe you can do them autonomously. The challenge is that because as the sample of water is pushed through it, these objects will actually move. So over here, you have a histogram. On the y-axis is count of objects, and on the x-axis is how much those objects in the red circle move. They can move. They can be static, 0 pixels moved, up to 60 pixels moved. So you can't just say I can say everything that XY XY plane, I'm just gonna, that is still there from the previous object, I'm going to remove, right? You have to account for that drift or motion of an object that doesn't really belong in a sample. So what we did is we developed an hierarchical image processing approach that first clusters all the objects based on their XY location, and then subclusters each object in a specific XY location based on their shape to determine whether the ARTiMIS had seen that object in the prior sample. So what that means is if based on the XY location and based on the shape of the particular object, if I can determine that I have seen this particular object in at least the last 3 frames, what that means is I need to remove this object from further processing, because it doesn't belong in the sample. And that's what we did. So this is an example. I'm showing this as like a cartoon of an actual sample, and what you're seeing is these orange, all the black dots are real objects in this particular sample, and all the dots that jiggle around, the spheres that jiggle around and have color, they are background particles. So we figured out an approach to actually remove those background particles. It doesn't work 100% well, but it actually works really, really well. So for example, on the y-axis is background. So BPD rate means background particle removal rate versus distance, how much distance it travels. And on the x-axis is pixels. So as the object, as a background particle or a stuck particle travels farther and farther away from its original spot where it was detected, we are unable to remove it. But if an object kind of travels up to 22 pixels or so, we can successfully detect that particle and remove it from a sample. And it actually works quite well. We've done simulations with synthetic samples, so where we take an image and we add background particles synthetically. And we've done this experiment with real samples at different cell concentrations. And what you see in the blue bar is our background particle removal accuracy is actually quite high, even at low cell concentrations, in excess of 90%. As the cell concentration increases, we are really good at removing background particles. As the cell concentration decreases, some background particles we are not able to remove, but it's still, in terms of 90-odd% accuracy at low cell density, it is not bad. So this was the second success that I would say of this particular project, is how do we take a look at an image and tell if the particle or if that cell actually belongs in that particular sample and is not a leftover from a previous sample. And you can imagine why this becomes very, very important. Now, the next question that we wanted to ask is, what is the ARTiMIS looking at? So here in this image, you have a cat and a dog. If I ask you what's in this image, if you are on the left side, the red says this is what you're looking at, you would say this image has a cat. And if you're on the right side, you would say the image has a dog. So it matters where you're looking, right? Where you're looking in the image makes a big difference. So when you take these images and you feed it into a machine learning algorithm, you want to know where the algorithm is actually looking at, right? So I'll take this and I'll take 2 samples, 2 images over here. So in this image, as the color gets red, that means our deep learning algorithm is basing its predictions on the areas that are colored red. And in these 2 samples, yeah, it's my cells that are colored red, and stuff where my cells is not present is colored in blue. So my image processing or deep learning algorithm is actually using the right information. Unfortunately, in this sample, and this is a microcystis cluster, nope, my deep learning algorithm is actually not using that microcystis cluster at all to provide any predictions. It's actually using part of the image where the cell is not there. So we had to actually painstakingly sort of go through lots and lots of model architecture optimization so that the deep learning model, so we could assign a prediction score based on where the algorithm was getting its information from. So if the probability score was low, then we knew that even though it might be predicting, oh, this is cyanobacteria, or this is an aphinozomenon, that maybe that prediction is coming from part of the image where aphinozomenon really doesn't exist. if that prediction score is high, then maybe it's actually coming from part of the image where there is a cell. So this becomes a really important point. We want to be able to sort of validate our predictions are based on the right kind of information. So this was a third really important aspect of this particular project, in that is the deep learning algorithm making a prediction based on the right information? And not a lot of classification approaches are actually, or image processing approaches are really paying attention to this particular aspect. The next question was, can the ARTiMiS process the data? Now, I know the biggest selling point for us when we were looking at the HAB monitoring landscape was that it's relatively cheap. And what we didn't want to do, at least as a build, it's relatively cheap. And what we didn't want to do is get it to where we had to put in, construct such a complicated deep learning model that you need a high power computer. And that's not where we wanted to get to. We wanted to be able to run the trained model on the ARTiMIS itself, on the single board computer that's embedded within the device. So the next important accomplishment of this particular project is that we experimented with a wide range of models. And on the y-axis, x-axis, you have number of parameters a model is using. On the y-axis, you have what its classification accuracy was. And as the number of parameters that you're using starts increasing, either you have to drastically increase the amount of time the model takes to predict what it's seeing, or you're going to need a much more expensive computer. And that's so. If you take some of these off-the-shelf classification models that are online right now, you're actually looking at millions of parameters, and that's not what we wanted to do. We wanted to find a lightweight model that uses the least number of parameters, while at the same time providing the highest level of accuracy. And this is what we did. We played around with, as you can see, lots and lots of different model configurations to identify. And just because you have more parameters doesn't make a model more accurate. It's the structure of the model that matters as well. So we went through lots of optimization and iteration to identify that model structure that uses, maybe 100, on the order of 80,000 or so parameters, and brings us at very high level of accuracy as well. And when we use this model, and again, you're seeing what I'm calling a confusion matrix. On the y-axis is a human annotated label. On the x-axis is a model predicted label. We were able to achieve about 94% accuracy for these freshwater systems, despite the fact that the diversity is so high. So what I can report now is the ARTiMIS can process the data. You don't need a separate laptop. The challenge that we struggled with towards this project that remains an important challenge, and there are lots of people from the water sector here, and I think listening to this webinar, and hopefully I can make a case for why solving this challenge has to be a community effort. And the problem is the problem of unknown unknowns. And over here, what I have is, this is just a cartoon. I have class 1, class 2, class 3, class 4. These are classes that my model has seen. And then I have 2 clusters that my model has never seen. So as an example, these freshwater systems are hyper diverse. So it's very, very likely that you're going to run into a situation where you're seeing something under the microscope that you've never seen before. Now, what is the problem with that? The problem is that a deep learning model that is trained on these 4 classes will split the space into 4 classes and lump in the unknown classes with the known classes. What this means now, I've trained this model on the 4 known classes. I have 2 unknown classes that I've never seen, but they show up in a sample. And suddenly my model is now saying, hey, you don't have 6 objects of class 1 in there, but rather you have 6 plus another 6 objects of class 1 in there. And that is not correct. That is inaccurate. And that could lead to, you can imagine, that could lead to a false alarm. What we want is to be able to get to a point where the model says, you know what, there are 4 objects that you've seen before, but there are 2 objects that I have never seen before. Get an expert to try to figure out what these are. This is a very, very important point, because the last thing that we want in terms of deciding in terms of accessing our source water is uncertainty coming from harmless stuff, right? It could simply be something absolutely harmless, and suddenly it's lumped in with your microcystis class, and the microcystis concentration has now gone up significantly. That's what we want to avoid. So what we've developed, or what we are developing, is an approach. It's an open set recognition approach. So we are leaning on all the great advancements that are happening in computer science. What we work towards the end of this project is an approach called classification with rejection. Using probability thresholding. That means when a model predicts that this object belongs to a specific class, it also tells you how confident is the model that object belongs to the specific class. So on the y-axis, you have high precision of prediction. On the x-axis, you have high recall of prediction. And the higher the value on both sides, the better you are. Now, this is an example of precision and recall for a model for a whole bunch of different classes when the classes were known to the object. So we are in this high precision, high recall space. Now, as soon as I add objects to this image that the model has not seen before, this is what happens. Suddenly, the recall stops dropping, the precision starts dropping for those objects, and that's not what we want. So what we have developed is it's pretty logical. It's not simple. Is the model now takes in these particles, it goes to a classifier which is trained to identify what it knows and toss out particle that it is not sure what it is, that it is not 80% or 70% sure that it might be some of the known classes, but maybe it's not. And it creates a bin for manual curation. And here an expert comes in and says, is this a known class, an existing class, or is this a novel class? And this is where the human intervention comes in now. And a novel class then gets added back to the training data set, and it trains the data set. So we go through this iterative process of identifying what we know, what we don't know, make what we don't know a known object, and then train the model again. And this is an ongoing process. This is quite new, but what you are seeing is as we do classification with rejection, our model precision and accuracy starts going up. Now, please note that this is still not where it should be, right? It is precision of still not where it should be. But as we start accumulating more and more data, as we start iterating more, this green curve hopefully will start moving up towards this blue curve. So on this, I'm going to put an orange and not a green. Do we know everything? No, we don't know everything. We may never know everything. But if the model can tell us, can do some level of curation for us in terms of these are the objects that you've seen before, and this is stuff that I think maybe you have not seen before. Let's go take a deeper look into it. So what we've developed with this particular project is, one, we've adapted the ARTiMiS to be used in for freshwater systems. Second, we've developed a workflow that works for freshwater systems where you have high diversity and high variation in biomass concentration with objects that you've likely never seen before. What we would like to do, so I will say that we have gotten the ARTiMiS somewhere around the 7 technology readiness level. Getting it to the 9 means that we need to play around with this classification with the rejection approach for a little bit of time. So that is where I think there is going to be a significant amount of effort that has to go in to get it from here to here, right? And this is where our utility partners, our water community can really help us kind of take it from technology readiness levels 7 to 9. With that, I'm going to thank the entire research team. None of this would have been possible without the entire team's contribution. And if you would like to collaborate, if you would like to follow up with questions, You can go to our project website, which is called phycosite.com, and you can share your details, and we can that way we can kind of stay in touch. We can send you updates on how our instrumentation is developing, how the deep learning tech is developing, and also if there are opportunities to collaborate, please do drop in a word over there. With that, I want to thank you very much for your attention and your questions, and I'm excited to answer them all.
Thanks, Amit. We have a lot of great questions. So I think what we're going to do is start with kind of a rapid fire on some of the logistical questions, and then we'll save some of those larger future-oriented ones for the end. So to start, does ARTiMiS have an output to SCADA?
So the ARTiMiS right now, it'll output the data onto a sort of a USB stick or SD stick, but that is not a challenging thing. So we can output it to a computer as well. We just haven't had an opportunity to kind of hook it up to a SCADA system, but it's just like a pH sensor. It's not going to be that complicated.
Great. Next question, does this work for high concentration samples and with samples with a variety of other samples with high turbidity?
Yes. So we have run fairly concentrated samples through that. And so yeah, we haven't yet. In fact, we've even run samples that were that were run through the plankton net, so really high concentration. So that hasn't been a challenge. One thing that I will say is we still rely on light transmittance through the sample. So if the sample is so turbid that the light can't transmit through the sample, then unfortunately it's not going to work very well. So we still rely on certain level of light transmittance. Yeah,
that makes sense. Does the pumping distort the shape or structure of the zoo phytoplankton, thus impacting the identification?
Okay, that's 100% amazing question. So we have not observed that. Our pore sizes are large enough, and the pumping is relatively passive enough to where we don't see large any sort of breakup of microcystis colonies. You saw, for example, those large colonies that pass through there. So we have not yet observed it. It's about the pore sizes that we're talking about is about 200 microns, a bit larger as well. So the chip that we also image in. So we have not yet noticed this. I'm not going to say that is 100% impossible. Maybe that's an interesting area to kind of investigate for us. We have not yet observed it.
Okay, and tied to where I think you were going on that one, can you describe the size of the algal cells you were working with?
Size of the algal cells, I think, so the smallest size that we can get to is about 3.5 microns. We can detect down to about a 0.5 micron, but we don't really see any features, the algal cell features or bacterial cell features at that size to really do much, except say that something's there. So anything above 3.5 micron is fine, and we've gone up to a couple hundred microns or larger, no problem.
Okay, great. Do you calculate biovolume of cyanobacteria?
Amazing question. Right now, we do not. Right now, we can get count, we can get surface area. What we are developing right now is a feature to where the ARTiMiS can scan along the depth of the chip, and we want to use that feature to calculate the biovolume. So we have not gotten there yet, but I know how important that particular parameter is, and hopefully we will get there in the near future.
The next question is referencing something from your presentation where you talked about recalling. In a little bit of a simpler term, could you describe what that means?
Yeah. So how do I put it? Let me. I'm going to try to say. I'm going to try to kind of. So it is if an object has seen a particular, if a classification algorithm has seen a particular object, how accurately does it put it back into that same class? So for example, if I see if the model has seen microcystis before. How correctly does it classify microcystis? Now, precision and recall both use what's called as true positive, true negative, or false positive, false positive definitions in here. So it's a formula that we use. But in the simplest term, if I've seen it before, how correctly am I kind of saying what it is?
Okay, that makes sense.
Yeah.
The next question is on QA/QC. Just what are the methods that you used, such as injecting known samples routinely?
So that's a really good question. So like I mentioned, we also did grew lab cultures of the type of stuff that we were seeing in freshwater systems. So we have lab grown cultures of microcystis, of mesozomenin, and a couple other cyanobacterial species that kind of are escaping my memory at the moment. We use those to both ground truth our concentrations. So make sure the estimates of, when I say concentration, I'm saying like surface area of the object, for example, or count of the number of cells, ground truth those. At the same time, ground truth, how should we say it, what the taxonomy was. What I will say over here, and this was a unique challenge that we faced relative to the photobioreactor system. Some of the stuff was so diverse that, we weren't able to assign it a taxonomy. So what we essentially assigned it is a morphotype, because we were saying, we are seeing this type of object over and over again, but we can't give it a taxonomic label. So we gave it a morphotype, filament number A or filament number B, because it was just too hyperdiverse. And so that's where I think we still have lots to do to improve. Yeah.
This project did a great job of curating a photo library. So we've got a couple questions on how long did that take? What would it take for another utility to kind of develop that to make something like this run properly?
Okay, that's an awesome question. So initially, it did take a while. And I should say that when we started this project, we were also kind of finding our way through how to optimize the process really well. So initially, it was a process of, I take objects and I manually give it a name to create the photo library, and I use that to train my algorithm. But later on, we realized that we could label 10 images and then use those 10 images to create a model to fish out 100 more. So that way now what we've done is we've created a strategy of where creating your training data set becomes a lot simpler and a lot faster. So you don't need a human sitting. If you need 1,000 images in your training library, you don't need a human to annotate all 1,000. You can use 10, fish out 100, then take a look at those 100, throw out the 10 that don't belong there. Then you can use the 110 or 100 to now fish out 1,000. So the process has become quite a lot cheaper in terms of time.
Great. And kind of on cost, do you anticipate that the cost is going to increase as additional work is completed in order to refine the quality of the output?
Yeah, that's a great question. So I don't have a good answer on cost. I can just tell you what it takes us to build it right now. But if you have questions related to it, please do send an email, and that's fine. And I'm happy to chat a little bit more to see, for example, what would be the nice price point for this technology for our utility community? Yeah.
Great. And I know the answer to this next one, but I think it's important that everyone else has a very explicit answer. So do you believe that ARTiMiS will only work within this specific watershed due to the form of the HABs, or can it be applicable to other watersheds?
That's it's a very good question. So if you were to use our training data set that we have right now, you probably are limited to the environments in which these objects exist. These cyanobacterial species exist. It's quite wide, but it still does. But the fact that we have made we have developed a framework means, and we've gone through all that optimization means that you don't have to go through this optimization. The time that we spent the last 3 or 4 years actually developing that workflow means that the ability to take the ARTiMiS and apply it in a new setting, the amount of time it's going to take is compressed quite significantly. So just as an example, creating the training library took us months and months at the beginning. It took us, but now I think maybe a couple of weeks, and you could have an initial training library for your watershed. So just the fact that we ran into these challenges and then we were able to solve them with a certain level of success means that our utility partners, our utility community doesn't need to run into the same challenges again.
That's great to hear that others can take the lessons you've learned to build their own.
100%.
Okay, so now we've got a little bit of a technical. So on the issue of counting microalgal cells in a cluster due to autoflocculation of individual cells, why not treat the sample to cause the microalga to deflocculate, perhaps to change the pH?
Fantastic question. And I think in the lab scale setting, that is absolutely fine. One of our original motivations of something like this was that there should be no consumables involved. So we don't want an operator to have to go and fill in a vial of something on every so often basis. The other thing is that once you start adding something to a water sample, you can't put it back into the environment. Now it's a laboratory waste, so you have to deal with it. So those are two reasons why we kind of there is a lot of value in doing it in the lab, 100%, but we got into this project thinking about it as a field application, so we didn't want to get there. The second thing that I will say is we are realizing this over the last year or so, not only in this project, in other projects as well, how these algal cells or phytoplankton aggregate can tell us something about the environment. It actually catches a physiological response that the algal community is going through. And that physiological response could be incredibly important as an early warning detector. So for example, if we suddenly started seeing microcystis colonies that are 10 times larger than before, and we only see this because we're not dispersing them, that is a useful piece of information to have. That means something is happening now that wasn't happening, and should we pay attention to this? So two reasons. One, we wanted to avoid any consumables or creating any hazardous waste out in the field. And three, we think there is actually lots of value in taking a look at things just as they are in the environment.
Okay, I think we have time for maybe one or two more questions. So how do you evaluate what is considered low, moderate, and high density?
That's a really good question. So in the report, we have precise concentration values. So the way in there, and I don't remember them right now, but the way we classify them in low, medium, and high is the ARTiMiS is looking at a certain field of view. It is looking under the microscope, and we classify it as low, medium, and high based on what % of that field of view was covered by specific objects. So for example, and I'm just going to throw numbers out there because I don't remember this. It is in the report. So if 5% of the image was filled with cells, maybe we call it low. If 30% is filled, we call it medium. If 60% is filled, we call it high. So I think it's something like so that's how we're categorizing it. If you want to see the precise concentrations there in the report, I'm also happy to kind of exchange emails and share that information with you.
Okay, I'm going to ask one last question before we run out of time. Have there been any attempts to use the ARTiMiS on mixed liquor of activated sludge wastewater treatment plants or to count microplastics?
First question, in terms of mixed liquor, no. I think this is where the concentration is probably a bit too high, for it to work quite well. I'm not going to say no, but we really haven't tried. So if you're interested in trying, please reach out. I'm happy to kind of get into that conversation. Microplastics, this is an important one. This has been brought up multiple times now. If the Water Research Foundation wants to fund a project looking at microplastics in the ARTiMiS, we are game.
Okay. Well, with that, I want to thank Dr. Pinto. This has been fantastic. We still have a few questions. I'm sorry to everyone that we didn't answer. We will follow up with you afterwards. I'll make sure I pass on your questions so we can get answers for you. But thank you, Dr. Pinto. This was really wonderful. Thank you to our audience for sticking with us through the end. I hope you learned as much as I did. A recording of today's presentation will be available after this, as well as the slides. So feel free to rewatch it, to share it with your colleagues. You can also access the project's report on the page now. And lastly, just please stick around for one last poll to help us gather feedback on the webcast. With that, everyone, have a great rest of your day. Thank you.